Welcome back to the conversation. I'm Nexus, your host. You are listening to the Agent House Conversations. Today, we're talking about a word that everyone uses, but not everyone means in the same way: intelligence. And we're tackling a question that gets heated fast. Are observed differences in intelligence between individuals and groups primarily driven by genetics, environment, or the way the two interact? Joining me to parse this out are two distinct voices. First, our Genetics Primary Proponent, who argues that stable differences are largely rooted in DNA. And counter to that, our Environment Primary Proponent, who argues that opportunities and life conditions are the strongest levers for potential. Basic ground rules: keep it civil, no personal attacks. And crucially, if we use acronyms or insider terms, we define them in plain language as we go. First, a simple definition so we're not talking past each other. IQ is a score from standardized tests assessing human intelligence, normalized to a mean of 100 and a standard deviation of 15, as typically defined in psychology. When we say IQ, we're talking about that specific measurement outcome, not someone's character or human worth. You'll also hear the term G factor. This is a psychometric construct, the shared variance or broad mental capacity that underlies positive correlations across diverse cognitive tests. In plain English, people who do well on one kind of thinking task often do well on others, and G is the statistical summary of that pattern. One last thing: primarily driven is a trap. It can mean what explains differences between individuals right now, or it can mean what causes group averages to differ. Those are not the same question. Genetics Primary Proponent, I'll start with you. Open this up for us. What is your baseline view on what drives these differences? And are we talking about individuals or groups?
Thanks, Nexus. You laid out the definitions of IQ and G perfectly, so I won't rehash them, except to emphasize that G is the signal we care about most because it reflects that broad, shared mental capacity. To answer your question directly, my baseline is that while environment clearly matters for whether a person thrives or suffers, genetics explain the bulk of the stable differences we see between individuals in modern societies. When we look at why one sibling struggles with complex reasoning while another excels, despite sharing a home and parents, that variation is substantially rooted in biology. It is important to clarify what I mean by genetic here. I don't mean there is a single smart gene. Intelligence is a highly polygenic trait, influenced by thousands of genetic variants, each with small effects, as shown by genome-wide association studies, GWAS. It's a lottery of thousands of tiny weights. Regarding your point on groups versus individuals, I think we have to treat them as separate questions. The evidence for genetic influence on individual differences is overwhelming and precise. The causes of group differences are much harder to disentangle from history and environment. I want to be careful not to confuse the two, but we also can't pretend the biological reality of the individual disappears just because comparing groups is politically sensitive.
You've drawn a sharp line between individual variation and group averages, a key firewall we'll need to watch closely. Environment. Primary Proponent, I want your opening view. Does environment just mean schooling, or something deeper? And do you accept that biological signal as the baseline we're building on?
To answer your question directly, I absolutely accept that there is a biological signal. We are biological beings, and no one seriously argues that genes play zero role in the brain. But environment is much more than just sitting in a classroom. It encompasses everything from the nutrition that builds that brain to the toxins that might damage it, and the cultural software we load into it. The problem with focusing so heavily on stable genetic differences is that it treats intelligence as a single, fixed number. We need to distinguish between two things. Fluid intelligence involves abstract reasoning and novel problem-solving independent of prior knowledge, peaking early in adulthood. while crystallized intelligence refers to knowledge and skills built through education, culture, and life experience. It remains malleable and responsive to environment across the lifespan. My counterpart is right that genetic differences might predict who ranks where in a specific group. But if we care about what drives outcomes, meaning how much potential is actually realized or how to close gaps between groups, that is primarily an environmental question. You can have a stable genetic rank, but if you change the environment, you can shift the entire distribution of performance upward. That leverage is what I'm interested in.
All right, let's do the family evidence, because this is where people throw around the word heritability, and half the internet talks past itself. Heritability is the share of differences between people in a specific setting that's associated with genetic differences. It's not how genetic you are, and it doesn't tell you what would happen if you changed schools, nutrition, stress, or opportunity. A twin study compares identical twins to fraternal twins, and an adoption study compares resemblance to biological relatives versus adoptive families. Both are trying, imperfectly, to separate genetic similarity from the rearing environment. Environment Primary Proponent, I'll start with you. What do you think is the strongest, honest takeaway from twin and adoption studies? And where do you think people routinely overreach?
The honest takeaway is that within stable, decent environments, twin and adoption studies consistently find that genetics explain a meaningful share of the differences between individuals. If everyone has enough food and schooling, genetic differences become the main reason people vary. But the massive overreach happens when people treat heritability like a fixed biological constant. It isn't. It changes depending on the quality of the environment. A classic example of this is the scar row effect. Turkheimer et al. 2003 reported near zero IQ heritability, h² approximal 0.10, and approximately 60% shared environment variance in low SES 7-year-old twins. In plain English, when life conditions are harsh or chaotic, the environment becomes the bottleneck, effectively suppressing genetic potential. High heritability is often just a sign that a society has equalized opportunities enough for genetic differences to actually show up.
That's a crucial distinction. Environment as a bottleneck. Genetics Primary Proponent. If heritability can drop to near zero when conditions are tough, doesn't that suggest that for a huge slice of the population, environment is the primary driver? How do you reconcile that with your primarily genetic headline?
I'm not allergic to that finding at all. In fact, it fits perfectly with the idea of gene-environment interaction. When resources are scarce or chaotic, the environment acts like a volume knob, turning down the expression of genetic potential. But that doesn't mean the genetic signal isn't there. It just means it's being suppressed. The real test of primary driver is what happens when you look at adults who didn't grow up in extreme deprivation, or who were raised in entirely different environments. This brings us to the most striking evidence we have. The Minnesota study of twins reared apart found a monozygotic twin intraclass correlation of 0.72 for IQ in adults reared apart. Bouchard et al., 1990. These were identical twins, separated early and raised in different families, schools, and neighborhoods. Yet as adults, their cognitive scores were nearly as similar as twins raised in the same bedroom. That degree of similarity is extremely hard to explain if the environment is the primary architect of our stable differences.
Today we're getting concrete about modern genetics, the studies that scan the genome, and the polygenic scores people build from them. This is where the conversation often gets overheated, because the numbers sound impressive, and people want them to answer questions they can't actually answer. Quick definitions so nobody gets lost. A GWAS, a genome-wide association study, is a research method that scans across the genome to find small statistical links between genetic variants and a trait. A polygenic score is a single number that adds up thousands of tiny genetic effects from a GWAS to estimate someone's genetic propensity for a trait. And R² is just statistics speak for how much of the differences between people the score can actually account for. Here's the key tension we're going to test. Within the same ancestry and similar environments, polygenic scores for intelligence can predict a meaningful slice of variation. Recent meta-analytic work puts that at roughly 6 to 8% of IQ variance in independent European ancestry samples, with some reports slightly higher. But once you try to carry those scores across ancestry groups, predictive power often drops sharply, down to 1 to 2% in some cases. Genetics Primary Proponent, I'll start with you. When you say polygenic scores predict IQ, define that for listeners. What does that 6 to 8% actually represent, and what does it not mean?
When people hear prediction, they often think of a DNA test that tells you exactly who you are, like a blood type. That is not what this is. A polygenic score is more like a weather forecast or a loaded die. It gives you the probability of an outcome, not a guarantee. If you have a high score, you are statistically much more likely to find complex cognitive tasks easier, but you could still struggle if the environment is hostile. But we shouldn't downplay the size of that signal. While the specific number for IQ is around that 6 to 8% range you mentioned, the signal gets stronger when we look at the broader picture of cognitive development and schooling. A 2022 GWAS meta-analysis of educational attainment, and about 3 million, mostly European ancestry, produced polygenic scores explaining 12 to 16% of variance, up to 15.8% in independent European ancestry samples. The scores also predict cognitive performance measures. What that tells us is profound. Thousands of tiny genetic variants are exerting a real, measurable push on how people learn and reason. It proves that the biology of intelligence isn't just a theory we inferred from twins, it's something we can actually measure in the DNA, even if it doesn't determine a person's fate.
You're leveraging education scores to talk about cognition, which opens a door. Environment Primary Proponent. If education and intelligence share so much genetic architecture, correlations often cited around 0.70, doesn't that suggest the schooling effect you care about is partly rooted in the same biology? Or is that education score measuring something else entirely?
I agree that GWAS show genetic correlations of 0.70 to 0.80 between intelligence and educational attainment, indicating substantial shared genetics as of 2021. But we have to be very careful about what that shared signal actually means. Education is a social outcome, not just a biological one. When a polygenic score predicts educational success, it is often capturing dynastic effects. The fact that parents who pass on certain genes also tend to provide environments filled with books, stability, and better schools. In those cases, the genetic score is effectively taking credit for the environmental advantage the child inherited. This is exactly why these scores are so fragile when you try to move them across populations. Polygenic scores for intelligence have reduced predictive power, R² often 1 to 2%, outside European ancestry groups, mainly from genetic architecture differences like allele frequencies and LD. If the tool breaks the moment you step outside the group it was built in, we cannot responsibly use it to explain differences between groups.
Now let's talk environmental malleability. How much measured intelligence actually moves when living conditions change? Quick definitions as we go. The Flynn effect is the pattern where average IQ test scores changed across generations, often rising over the 20th century, suggesting something beyond genetics is moving the needle. And IQ points are just score differences on standardized tests, useful but not a complete picture of a person. To anchor this with real numbers, researchers describe the Flynn effect as generational IQ increases averaging about 2.3 to 3 points per decade in developed nations during the 20th century. And in one classic dataset, Dutch conscripts' Raven scores rose 21 IQ points from 1952 to 1982, as James Flynn reported in 1987. Environment Primary Proponent, the opening question is yours. What do you think was actually changing in the environment that could move scores that much? And is it a lever we can still pull today?
The sheer speed of these changes is the most critical point. Genetics simply cannot evolve fast enough to explain a 21-point jump in 30 years. That tells us definitively that environmental factors, like nutrition, reduced pathogen burden, and dramatically increased cognitive stimulation, are powerful enough to shift entire distributions. And crucially, this isn't just a story about scores going up. They can go down too, which proves responsiveness. Norway saw a Flynn effect reversal in IQ scores, about a 7-point decline per generation for cohorts born after 1975, and a 2018 PNAS study using within-family comparisons confirmed this was driven by environmental factors, not genetics. This matters because it identifies the levers we can actually pull. For example, iodine supplementation in iodine-deficient regions increases average IQ by 8.7 to 13.5 points, according to meta-analyses. That is a massive, causal, biological change driven entirely by policy, proving that when you fix the environment, you unlock potential that was previously invisible.
Iodine is a strong example of fixing a biological deficiency. Genetics Primary Proponent. Do you view that kind of deficiency correction as a special case? Or do you accept that environmental floors, like nutrition and health, can meaningfully move population averages, even if rank order remains heritable?
I absolutely accept it. When you fix a severe biological constraint, like iodine deficiency, you see massive gains because you're removing a brake on development. That is a clear win for environment. Removing damage recovers potential. But there is a huge difference between fixing a deficit and indefinitely raising intelligence in healthy populations. The Flynn effect shows we can lift the population average, but it doesn't explain why, in a classroom where everyone is well-fed and schooled, some kids still struggle, while others excel. The mean moves, but the stable differences between individuals remain. And when we try to push beyond those basics in developed settings, the gains often don't stick. Preschool showed 10 to 11-point IQ gains at program end, age 5, which faded by ages 8 to 10 per the study's data. That fade-out is the crucial reality check. Removing environmental harm is distinct from permanently boosting general reasoning ability.
Now let's go to the hardest part of this topic: group differences, and what, if anything, we're supposed to do about them. I want this to be a careful conversation, because people often confuse 3 different questions: what predicts outcomes, what causes outcomes, and what policies are fair or legal. Also, quick definitions for listeners. Heritability is about differences within a group in a specific environment, and gene-environment interaction means the environment can amplify or dampen genetic effects. Those 2 ideas can both be true at once, and that's why this debate gets messy. Let's start with persistence. If some group gaps remain over time, what's your best explanation, and what would you predict changes them fastest? Genetics Primary Proponent, you first.
Before we go into group gaps, let's set a ground rule. We are talking about statistical averages,
not about what any individual can do. No person is reducible to a group mean, and you can have overlapping distributions even when averages differ. But to answer your question on persistence, if the environment were the primary driver of these gaps, you would expect them to close significantly when children are raised in highly supportive similar home environments. Instead, we see stubborn stability. The 1992 follow-up to the Minnesota transracial adoption study reported mean IQs at age 17 of 89 for Black adoptees and 106 for white adoptees. This pattern, where changing the rearing environment doesn't erase the gap, is consistent with a model where genetics is a major driver of stable differences. So while we should absolutely fix environmental harms like lead exposure or bad schooling to raise the floor, we shouldn't promise the public that equalizing opportunity will necessarily make all group averages identical.
That Minnesota study is often the flashpoint in this debate. Environment Primary Proponent. When you hear that result, do you see genetics, or do you see other mechanisms, like social treatment or measurement issues, that adoption doesn't erase? And what environmental levers do you think could actually move a gap that stubborn?
I see a study that is informative, but not a verdict. Adoption changes the home, but it doesn't equalize peer groups, identity, or the way society treats a child outside the front door. It doesn't erase discrimination or stereotype threat. More importantly, if we want to explain why gaps persist, we have to look at how environment moderates potential. The Scar-Rowe hypothesis states that IQ heritability tends to be lower among children from low SES backgrounds due to greater environmental variation, as shown in some twin studies like Turkheimer, 2003. That means in tougher environments, realized intelligence is suppressed by the setting itself. So the lever is obvious: fix the suppression. A 2018 peer-reviewed study in Rhode Island found lead exposure disparities accounted for 1 to 2 points of the Black-White test score gap, with urban remediation policies producing lasting cognitive gains. That's not a magic wand, but it's a concrete, scalable lever that reduces inequality by improving the physical world children grow up in.
This is where the argument stops being abstract: hiring and promotion. The Supreme Court's 1971 Griggs v. Duke Power decision created the disparate impact standard, limiting use of job tests that screen out protected groups unless proven job-related. But in 2009's Ritchie v. DeStefano, the court ruled 5-4 that New Haven could not discard firefighter promotion exam results just because of racial gaps. Put those two together. What do you each think the law is trying to balance here? And is it doing it well?
I support fixing lead pipes, absolutely. But we have to look at the scale. Remediation might close the gap by 1 or 2 points, which is good, but the US Black-White IQ gap has remained around 1 SD, 15 points, since the 1970s, despite some socioeconomic progress, though recent data shows slight variations. We are dealing with a much larger, more stubborn difference than a single environmental fix can explain. The law is stuck because it's trying to reconcile two things that conflict in the real world: meritocratic selection and equal group outcomes. Griggs allows tests if they are job-related. The problem is that cognitive ability is highly predictive of job performance, so valid tests will produce disparate impact as long as those group differences persist. The courts are effectively asking us to pretend the difference is in the test rather than in the distribution of the trait, which forces a hard choice between competence and representative quotas.